Dear friends, in this video lecture, we will have an overview of the cell junction. One of the important characteristics of tissue is some cells which are close to each other, especially in the case of epithelial tissue, they need to be attached to each other to improve the functionality of the cluster of cells as a whole. Here you can see a photograph of a simple columnar epithelium, a single layer of pillar-like cells resting on a basement membrane. Adjacent cells are glued to each other. Those lines that are there in between the cells represent the fused cell membranes of two adjacent cells. But this level of gluing varies in different parts of the cell, and the characteristic of the adhesion depends upon the regional requirements between the two cells. Let's see some of the common types of cell junctions. It can be in a spectrum as wide as from a total barrier junction, that is the tight junction, to a system where there is a considerable gap between the cells that enable a more freer transfer of materials across the cell. The first one will be tight junctions. This is particularly characteristic of epithelia, especially like the transitional epithelium in the urinary bladder and the ureter, where the cell junctions are so tight and virtually plastered to each other, close to their apical surfaces, so that urine cannot percolate through it into the interior or the deeper layers of the organs. Now that is a very good example. Similarly, you can see these kind of tight junctions in epithelia, like in the case of the stomach mucosa, where the acidity that is there in the lumen does not uh, damage the cell and go into the deeper layers of the, so therefore this is an excellent protective mechanism. Similarly, you can see this in the intestines also, close to the apex of the cell. Now let's see how exactly these tight junctions are designed. Basically, at the tight junctions, the adjacent cell membranes of the two adjacent cells are virtually plastered or touching each other, and sealing this like a welding system, sealing this junction are a group of proteins known as transmembrane proteins. Now those are the adjacent cell membranes. There's hardly any gap between the cell membranes, and the strands of transmembrane proteins will help in virtually sealing off or completely gluing these two cell membranes. And here you can see a small, very little gap between the two cell membranes, in between the transmembrane proteins. There's very tiny intercellular space may be seen. But this space is of no consequence because the whole purpose is total barrier protection. The, the next variant of this is the gap junction, where you will see once again the adjacent cell membranes shown by the two yellow arrows. Then at certain points the cell membranes come close to each other, but there is still a gap between the two cell membranes. This is very important. And connecting the cell membranes, the two cell membranes, is a tunnel like architecture. Now that tunnel is made up of a protein known as connexons. The the protein material that forms it is the connexin protein. So there will be such tunnels connecting, or like bridges connecting the two cells, especially in places where the two cell membranes are closely opposed to each other. So this arrangement makes sure that it is not a complete barrier, but it is a selective barrier, discretionary barrier. That means certain substances can go through this tunnel from one cell to another. Therefore, this is an example of a facilitated transport across the cell membrane. Now that's the intracellular space. We will next go into another type of junction where the cells go into a kind of an adherence to each other. Now you see this junction is known as anchoring junction. It's just not that the cells are just attached to each other. These cells are reinforced. The points of attachment are reinforced by rivet like mechanisms, which are once again membrane proteins. Now for example, this one, desmosomes, macula adherens. This is quite commonly seen in the epithelial cells of the skin, where the in the process of keratinization, that during that process these cells are virtually attached to each other, and slowly as they move to the surface these cells become flattened. But in the immediate stage after the basal lamina, that is the basal cells, then the basal cells, as they move towards the surface, you will see these kind of desmosomes very prominently. Now you see here what happens is the adjacent cell membranes are shown. Next, there is on the inner aspect of the that is on the cytoplasmic aspect of the cell membrane, you will see a proteinaceous material which resembles like a plaque or a shield. Now there is a shield on one side of the basement membrane of one cell. Similarly, there is a plaque on the other side. Connecting these two plaques, that means there are some kind of a screw or some kind of a material proteinaceous in nature, connecting or fixing these two plugs. Often you will see that this material that connects these two plugs, let's name them now. They are the transmembrane proteins called cadherins. They move from one plug through the intracellular space and into the opposite cell membrane, pierce this opposite cell membrane, and then anchorage. That means there is a cross anchorage between the two cell membranes using these rivet like mechanisms. And of course, because we are describing the example as a skin, there are filamentous proteins attached to the cytoplasmic aspect of the plaque. They are usually intermediate filaments, the best example being keratin. And of course, that's the intracellular space. And now you see, we will identify the intermediate filaments, namely the keratin in this particular case. We move on to the next example of anchoring junction. There are two types. One is the zonula and the macula. We have finished the macula. The zonula is virtually a replica. Here also there is a plaque on both the cell membranes on their cytoplasmic aspect. The only difference is the protein that is attached, the anchoring protein that is attached. Remember, this transmembrane protein cadherin is also the same. Instead of intermediate filaments, here actin filament will be present. I repeat, that is the only change between the macula and the zonula adherens. Now, there is a last category, fifth category, called hemidesmosomes. Here, the anchoring protein is not cadherin. Here it is integrin, and it doesn't plaster one cell to the other. Instead, it plasters the cell basal aspect of the cell to the immediately adjacent basal lamina. So these are the 5 types of cell junctions, the best encountered in epithelial cells. Now you see, to summarize, we have tight junctions, we have gap junctions, we have the adherence types where there is a macula type and a zonular type, and lastly we have the hemidesmosome type. Thank you. Thank you.